All right, we're going to continue. So, the scientific method has steps, and these are kind of it, but I want you to realize that science is not always just a straight line from observation all the way to conclusion. It can wind around, go back, mess up, restart. Science doesn't have one set path. In fact, if you were to Google scientific method, you would find other steps. Like some people include analyze, test, record results as different steps, whatever. I just think if you memorize this, you'll be pretty good. So if we just memorize, oh heck, we should be fine. All right, so let's begin with observation. Observation is not just seeing stuff. You can also smell it, taste it, hear it, touch it. It's any of those things. Once you make an observation, though, there's this thing called an inference, which you might not know about. An inference is like what you already know about what you observe. So, for example, if I hear a siren going down the road, that would be my observation. An inference is just something I already know. So, I probably already know that it's a cop or a fire engine or maybe it's a ambulance. So I pull over, right? Saves me some time in my decision making. Same thing with science, right? So if I have an observation that there's a bird and it has feathers, I might be tempted to make the dumb hypothesis, feathers make you fly. You probably already have inferences that help you to contradict that. Think of some contradictory inferences. Okay, so hopefully you realize that there are things that don't have feathers that can fly, like a bat. There are some things with feathers, like penguins and ostriches, that do have feathers but can't fly. This saves us some time so that we don't make dumb hypotheses. If we did make this hypothesis, we'd have to experiment and think about dropping an ostrich from a roof. That would be a tragic experiment. We wouldn't want to waste our time. So, inferences help us to make sure we make good hypotheses. So, what is a hypothesis? It's basically a statement that is trying to link cause and effect. So, it's a cause effect statement. We're trying to establish causation. It must be testable. You can't make a hypothesis like we are in a simulation, right? Because how would you test that? How could you even? That's not a scientific hypothesis. That's just a guess, really. Okay, and then it also must be disprovable, right? Or at least to be a good hypothesis, it must be disprovable. So you can't say something like, my grandmom's cookies are the best. I mean, they are, but that's my opinion, right? So your opinion might be different. My grandmom's cookies, she uses a lot of butter. They're really thin and chewy. I think that's the best. You might prefer fluffy cookies. Whatever. We can't disprove either of our opinions. Next would be full of inferences, like I said before. Helps us to save time, right? So here's an example of a hypothesis. If the sun is shining, then the grass will grow. Perfectly fine hypothesis. There is a cause and effect. In such a hypothesis, there is the cause and then the effect, right? So the cause and the effect are different variables. Variables are things that can change in an experiment. In this case, we have an independent variable, which is the sunlight. It is a variable that can change the other variable. So obviously, if there is sun, then grass grows. Right? So the cause here is sunlight, and the effect is grass growing. So the independent variable is the cause in your statement. So again, this was the hypothesis. The cause is the independent variable. Now it's important to remember you can only have one independent variable. Let's compare that to the dependent variable. The dependent variable is the variable that is changed. And you can have as many of them as you want. It's just something you measure, right? So I could test how long the grass grows, but I could also test how green the grass is, or how long it lives, right? So I can test multiple effects. Those are dependent variables, but I'm only allowed to test one independent variable. So we have to keep everything else constant. Constants are variables that we don't want to change. So say, for example, I have a bag of fertilizer. And I'd sure like to know if fertilizer can make the grass grow taller, right? I wouldn't want to test different amounts of sunlight and different amounts of fertilizer, because if I did that, what are these effects from? Are these effects the result of the sun or the fertilizer? I don't know, right? So we have to keep either the amount of sunlight or the amount of fertilizer the same. The one that we choose to keep the same is constant. So here's some examples of constants in this sunlight experiment. We would have the fertilizer, or lack thereof. We would have probably the type of grass would be important. Try to think of other things. What other things would we try not to test? Another example would be how much water we use. We want to use the same amount of water. And the area, where are we planting it? We wouldn't want to plant some grass in California and some in Virginia, right? So place, same place. These are all things that you don't want to accidentally make an independent variable. That's probably the biggest error I see in the scientific method on the SOL. Once you have your neat and tidy hypothesis that is testable and hopefully disprovable, you go to your experiment. In your experiment, you're going to test to see if it works or not, right? So in our sunlight experiment, we would have sun, and we'd have two groups, right? We'd have grass that is only out during the night. I guess we would have to use a shed or something, like a greenhouse where we could close it. And we'd have some in the day, and then we would compare them, right? And so hopefully grass grows tall in the sun and not very tall in moonlight. And that will give us our results. So that means we have to have parts. There is the experimental group where we have our independent variable, in this case sunlight, and our control group where there is no independent variable. And let me use another example. So let's say, for example, we're doing, we have a hypothesis. If you take antidepressants, then you will feel better, right? So how would I run this experiment? It's very obvious. You have two groups, right? And one, you give the antidepressants, and the other, you don't. And so you run the experiment, and hopefully this group feels good, and this group doesn't, right? Hopefully. But here's the problem. When it comes to medication, we need to be careful. Because this group right here, they might have felt better because they think they're supposed to, right? What do I mean? Have you ever accidentally had decaf coffee and you feel like you have more energy? It's because you think you took the real stuff, right? The power of suggestion is very, very strong. We call this the placebo effect. Another example would be, imagine you drank non-alcoholic beer and you started acting like an idiot. This is something that people do in fraternities. They prank their pledges. That happens, right? So same thing with medicine, right? If you take what you think is a real pill, you might feel better. So we have to account for this with placebo pills. So what we do in our control group, instead of just excluding the independent variable, we add a placebo. So we add a placebo but we also take away the independent variable. And so in this case, the independent variable is the medicine. In the placebo pill, we put sugar, and then we look at the results. If both groups have the same result, it was because of the placebo effect. However, if the placebo group does not have the desired effect, then we support our hypothesis. Okay, next, once we do our experiment, we record our data. And there's two types, qualities and quantities. Qualities would be like the dog is brown. Quantities would be like there are 3 dogs. Which do you think scientists prefer, numbers or qualities? Hopefully, you picked quantities. Quantities you can do more with. They're numbers. You can add numbers, subtract them, plot them, graph them, statistically analyze. You can statistically analyze them. And you can create what's called statistical significance. Statistical significance is very important in science. Say, for example, we did the antidepressant test, and we found that 99 out of 100 individuals felt better. That would be statistically significant. If we did that same test, however, and we found 50-50, like we found that 50 of them felt better, then that's not very statistically significant. So we want our results to be definitive, and we can test for how definitive they are with numbers better than with qualities. So one of the arts of science is taking a quality and turning it into a quantity. All right, sources of error. This is how we mess up in science. We've talked about skipping steps with anti-vax. You don't want to skip your experiment. Too many independent variables can confuse things, right? So in every experiment, there is cause and effect. The cause is the independent variable, and the effect is the dependent, right? If you have 2 causes on one effect, you don't know which one is actually causing the effect, right? So you need to only have one independent variable. Next is bias. We've talked about that. That is when you want an outcome, and so you tend to make it happen. You ignore data that does not conform to your beliefs. And then the next one is placebo. So real quick, let me talk about bias. Let me give you an example of bias. So say that you work at a oil company like Exxon, and you hire scientists to prove that global warming is not caused by man. No one is going to believe you because they know you have a vested interest in the outcome. That would be biased. It's a good example. The same reason why you would not want to believe a cancer study coming out of a tobacco industry. All right, next is placebo effect. We just talked about that. Believing that you will feel better, so you do. And then human error is just what it sounds like. It's when people mess up in experiments. All right, finally we have conclusion. There are 2 different types, inconclusive and conclusive. Once we look at our data, 1 of 2 things will happen. So if our data does not support our hypothesis, we just go back. And we keep going through the scientific method that way. We fail until eventually we discover something. Now, when our data does support hypothesis, we send it off to our peers, which are people that have a similar discipline to us. They try to repeat our experiment and do it over again and test to see if we're right. If we do manage to support, if we are supported by our peers, we might have a scientific theory. And so a scientific theory is kind of a big deal. You might have heard the word theory before and you think, oh, that just means like an idea. But a theory in biology is a big deal. So for example, the theory of evolution is something that we use as a foundation for other hypotheses. So a theory is like a foundation on which we can build new hypotheses. So for example, the theory of evolution supports the hypothesis that we evolved from chimpanzees. And maybe I didn't draw that very well. So this is the theory. It supports. It's really hard to write on this tablet. Have some sympathy for me. All right. So theories support hypotheses. They make them possible. So if you are a really good scientist, you might end up with one of those one day. I would show this video, but I'm kind of out of time. So anyway, I hope you liked it, and I'll see you.